Welcome to Discover ASAP, the video interview series where we highlight the top findings and tools connected to the ASAP network. Today, I'm talking with members from Team Edwards about their contributions to understanding mechanisms of levodopa-induced dyskinesia. So to get us started, Alexandra, can you talk to us briefly about Team Edwards' ASAP project and the impact that you hope to have on Parkinson's disease?
Great. So many people know Parkinson's disease as a neurodegenerative disease, a disease in which brain cells die gradually over time, causing people disabling symptoms such as movement and cognitive disorders. However, our team likes to think of Parkinson's not only as a neurodegenerative disease, but also a functional disease, meaning that there are alterations in the function of the cells that are still there in the brain, and that alteration in function, either the pattern of activity or connectivity within the brain, mediates many of the symptoms that people experience. And our team is interested in investigating how activity could actually accelerate or promote the neurodegenerative process, as well as mediate the symptoms of the disease. So the kind of combined role of activity within the disease.
Great. Yeah, I think that's a really important question. I think you're right that oftentimes when we think about Parkinson's, we focus a lot on the cells that are lost and not so much on the cells that are remaining and the kinds of maybe compensatory changes that are happening at the cellular and circuit level there. So as I said at the top of this interview, that we're talking about levodopa-induced dyskinesia specifically. And so I'm hoping that you can give me sort of a brief primer about what L-DOPA-induced dyskinesia is and why you think it's important that we understand it.
So many people who develop Parkinson's disease have motor symptoms or movement symptoms that are part of the difficulties that they experience, often slowing of movement and tremor and difficulty with walking, for example. And for this, they take what we call dopamine replacement therapy. These are medications that boost dopamine signaling within the brain. Levodopa or L-DOPA is one of these kinds of medications that almost everybody with Parkinson's takes at some point. And these medicines are very effective for some of the movement symptoms of Parkinson's disease. But after taking these medicines for a few years, people often develop complications, including levodopa-induced dyskinesia. Dyskinesia is involuntary movements, often dance-like, restless-looking movements that are triggered every time a person takes their medication. So the medication still produces a lot of benefits, but also produces involuntary movements. And this causes people to need to constantly adjust their doses, when they're taking, how much they're taking, in an effort to hit the sweet spot. And sometimes the sweet spot is literally unattainable. And so for this reason, people often have to cut back on their medication and eventually may resort to deep brain stimulation to manage dyskinesia because it can get in the way of the normal movements that they want to make. So it's a clinical problem that we don't really understand fully. Why do people develop it? Why does it get worse over time? And we'd really love to have some better tools to manage it for people.
Sure. So in order to better understand L-DOPA-induced dyskinesia, Alexandra, your lab, and Chris, your lab, are using animal models of dyskinesia primarily. So can you talk about the model that you guys are using to study this and how you're assessing dyskinesia in this model?
Yeah, so the main way that we go about it for the most of us in the field that are looking at this, to begin with, to mimic the loss of dopamine cells, we'll use a neurotoxin-based model. So we can lesion the dopamine cells selectively with a drug, 6-hydroxydopamine, and be able to lesion those dopamine cells to mimic the loss of dopamine neurons, sort of mimicking a later stage usually of the disease when most of the dopamine cells have degenerated. Then in order to mimic the dopamine precursor drug, the treatment of animals with levodopa, we'll administer those for a period of days, usually around 5 to 7 days, depending on the study. It can vary a little bit. And then over the course of this, the animals then at the end of this will then begin to initiate these involuntary movements over the course of treatment with the L-DOPA drugs. And so we'll do this for a series of days afterwards, after all the dopamine cells have degenerated. And then we can assess these abnormal involuntary movements, these dyskinesias in animals, by a series of metrics.
Great. So I think that leads us nicely into what I think will be the bulk of our conversation, which is, I'd love to hear from both of you about what your labs and Team Edwards sort of writ large have contributed to our understanding about L-DOPA-induced dyskinesia. You know, what have we learned about the mechanism and maybe possible treatment or intervention options for people who develop this symptom?
I guess I can start commenting on that. So I'm Emily. I'm a PhD candidate in Alexandra Nelson's lab at UCSF. And in our lab, we're interested in trying to understand some of these cellular and circuit mechanisms underlying LID. And like Alexandra mentioned, it's not fully appreciated what those are, but we're getting a better understanding in the field that maybe aberrant activity in the striatum is playing a critical role. And specifically, we found that hyperactivity in a subset of D1 dopamine receptor expressing medium spiny neurons might be a major contributing factor. And so when we think about what might cause excessive D1 MSN activity, we can think conceptually at three broad levels. Maybe there's changes in response to this loss of dopaminergic input in the case of Parkinson's disease, or maybe there's plastic changes with chronic treatment with levodopa on either the intrinsic excitability of these neurons, changes on the excitatory input onto these cells, or a loss of inhibition onto D1 MSNs. And so the project that I've most been involved in is focusing on that third category, thinking about a loss of inhibition onto D1 medium spiny neurons, which in this case we can think of like a removal of a brake. And so we normally think about the medium spiny neurons contributing to movement through their connections to downstream basal ganglia nuclei, but there's also connections, inhibitory connections between medium spiny neurons locally in the striatum. And because medium spiny neurons are the dominant population, about 95% of the cells in the striatum, we think that this has the potential to be a really powerful source for basal ganglia output. And so in our project, we used optogenetics and ex vivo electrophysiology to measure the strength of connections between medium spiny neurons in the striatum in healthy animals, Parkinsonian animals, and Parkinsonian animals that have chronically been treated with levodopa. And really excitingly, we found that these connections seem to be modulated by dopamine on both chronic and acute timescales. And so hopefully we can take this information, to your point, Dana, about how it might be relevant for treatment options. Maybe someone can take this idea that dopamine is acting or dopamine is affecting these connections locally, the microcircuitry in the striatum, in addition to the downstream projections. And so maybe translational teams could develop more effective treatment options by having more precise targets with fewer off-target effects. Dana, I think you're on mute.
Oh, yes, I am. Thank you. I think that is a fascinating finding, Emily, of this idea of the microcircuitry connections between the MSNs being altered in these Parkinsonian and dyskinesia having animals. Chris and Beatriz, do you want to talk about what you guys have found as well?
Yes, well, I'm Beatriz Nielson. I'm a postdoctoral fellow at CRISPR Lab. And well, following a little bit of what Emily was saying, even when we don't know exactly or we completely don't understand how the LID pathophysiology is, it is thought that there is an enhancement of that direct pathway in the striatum with an exacerbated signaling of this D1 signaling. And in our case, our project was mainly focused on looking for which alterations develop in the cholinergic signaling in the striatum, which is acetylcholine is the other most important neuromodulator in the basal ganglia and particularly in the striatum, and it closely interacts with dopamine. So we were particularly interested in that, and we found that there was a reduction in the M4-mediated modulation of that direct pathway in our model of Parkinson's disease. So then, as a follow-up, we were interested in what was going to happen as a consequence when those Parkinsonian mice start receiving the L-DOPA treatment and develop the LID. Because considering that in the LID, supposedly we have an enhancement of that direct pathway, and now we found a reduction in the M4 modulation, which is theoretically inhibitory, that lack of inhibition would be somehow facilitating that progression towards LID. So we basically examined that, and we found that it was like that adaptive response that we found in the cholinergic transmission was actually facilitating or accelerating the progression towards LID once the L-DOPA therapy was initiated. And also, as a part of that research work, we developed two rescue strategies to restore that M4 signaling that was originally reduced in the Parkinsonian mice. And we could see that those strategies were effective to also help to decrease or delay a little bit the progression towards LID. So I think that even when it's not directly a translational research, definitely it positions the M4 and the cholinergic signaling as a non-dopaminergic alternative target for these disorders. That can be, of course, as an adjunct to L-DOPA therapy and other dopamine replacement treatments.
I just want to emphasize that what's really exciting about this work, as well as work of several other teams in ASAP, is that traditionally people have thought about dopamine and dopamine signaling on the canonical cells within the striatum as being critical for mediating the positive effects of these medications, which they are. However, the work that you've just heard about is actually looking at other neurotransmitter systems besides dopamine, because that might be a way to maintain the positive effects of these medications while avoiding some of the complications by including tweaking plasticity, altering synaptic connectivity within the striatum. So these might be orthogonal strategies that could be really helpful eventually for therapeutics.
And I think I can also add to this. I think it's also nice to also see how a lot of the work that's being done sort of within our team, but also with others, really is complementary. So past work from Alexandra Nelson's lab had shown that there's subpopulations of medium spiny neurons that show enhanced dopamine receptor signaling, that it gets exacerbated, their activity gets exacerbated following levodopa treatment as well too. So we can think about our work as sort of framed within that as understanding that if this is sort of a pathological or a adaptation that arises when these cells from excessive D1 receptor signaling, these same neurons, this is where this muscarinic signaling is coming through. And so the loss of this is maybe then allowing for this runaway excitation of these dopamine, of these medium spiny neurons to occur. And so again, by allowing the dopamine therapy to have the positive effects, but also having these adjuncts coming in could be really beneficial. So I think we can think about a lot of our work sort of in the context of past works by many others in the field, including a lot of the work that's been done from Alexandra Nelson's lab.
Yeah, I agree. I think that talking about the work that both of your labs is doing, I think is really highlights the importance of collaborative science as well, because it's, you guys are building on the work that you are both doing sort of in separate labs, but towards, finding these complementary effects that hopefully get us closer to understanding these big questions in the Parkinson's field, which is exciting. Okay, well, I usually close my interviews by asking if there's anything else that you want to mention or talk about that you think is particularly relevant or important to the topic. And so if so, this is your chance. And if not, then I will close us out.
I just want to highlight how important it is that young scientists like Emily and Elizabeth join the field and are doing such incredible work and bringing their own creative minds to this. And this kind of work is supported both by ASAP and federal funding at this point in our country, and it's a wonderful thing.
Yes, I agree. We're very, trainees are the backbone of research in many ways, I think. So, all right, well, thank you guys.